It's my pleasure today to introduce Dr. Ronny Drapkin from the University of Pennsylvania. Ron received his bachelor's degree from Brandeis University, his MD PhD from Rutgers, and then was a resident in anatomical pathology at Brigham and Women's Hospital in Boston. He then was a postdoctoral fellow in cancer biology at the Dana-Farber Cancer Institute, and then was an assistant professor and then associate professor for about a decade at Harvard. Just recently, in 2015, he became the director of the Ovarian Cancer Research Center at the University of Pennsylvania, Perelman School of Medicine in Pennsylvania, and also the director of gynecologic cancer research. He seems, if you read over his resume, he's somebody who's been at the right place at the right time, was my assessment. He started out doing really general transcription factors and DNA repair. That turned into work on DNA repair in hereditary cancer syndromes, looking at Brca mutations. He then went into work on ovarian cancer and early detection. And some of you may be aware that, in ovarian cancer, it's the CA125 is the marker if you're following a woman for recurrence of ovarian cancer. But his work was looking at another protein called HE4, and he really pushed that to the forefront, and now it also is FDA approved to look for recurrence of ovarian cancer. His most recent work, which I think he'll be talking about today, is on ovarian cancer in the fallopian tube. And the idea here is that it's a new paradigm, which probably now you think it's an old paradigm, but it's basically the idea that ovarian cancer arises from the fallopian tube instead of the surface epithelial of the ovarian, of the ovary. He's trying to develop a number of novel model systems, which I think he'll be telling you about today, looking at fallopian tube cell line models, genetically engineered mouse models, and also patient-derived tumor xenografts. So he's got a lot to tell you about today. As director, though, of the Ovarian Cancer Research Center, he has the goal of trying to translate all these important biological principles that they discover in the laboratory into the clinic. So he leads a multidisciplinary team, and I think he'll tell you about what they're up to next. Thanks a lot.
All right, thank you, Amy, for that wonderful introduction and the invitation to come and visit. I look forward to spending the day today meeting a lot of the investigators and talking about some of the exciting work you're doing here. I will, start with my disclosures. Unfortunately, I don't have anything exciting to disclose. I just sit on a couple of consulting companies that don't pay me. I will talk a little bit about the use of CDK inhibitors in my presentation later on. So as an overview, I'm just going to talk a little bit about ovarian cancer, some statistics you're probably all familiar with, tell you somewhat about this new paradigm that Amy mentioned in terms of our understanding of its pathogenesis and the genetics that has evolved through the studies of the Cancer Genome Atlas, how that really led us to develop new model systems and how we're trying to leverage all that to really figure out how to best attack ovarian cancer. So let's start with the pathogenesis and genetics. So these are numbers, again, that you're very familiar with from the WHO. We know that there's about 250,000 new cases of ovarian cancer diagnosed worldwide every year. And unfortunately, the vast majority of these women succumb to their disease within a year or so. That's mostly because they get diagnosed at advanced stage 3 or 4, where the fiber survival really hasn't changed or improved in decades. And this is, for the pathologist in the room, a specimen we actually, I think, resected when I was a resident. I didn't do it, but one of the fellows did. So this is uterus, and this is a big ovarian adnexal mass. And that's unfortunately what happens, that these tumors can grow rather large before they make it out of the pelvis, causing symptoms that unfortunately themselves are not very specific. This makes ovarian cancer the most lethal gynecologic malignancy, and unfortunately we still don't have tools for the early detection of this cancer. Now, another stat I'd like to point out here is what I call the lethality index. So if you look at the number of deaths per cases, right, you find that ovarian cancer falls right behind pancreas and lung in terms of the lethality index at 0.67. And I'll just mention down here that even though breast cancer is about 10 times more common than ovarian cancer is about 3 or 4 times more lethal than breast cancer. Now, complicating all this is the fact that this is a heterogeneous disease. Much like breast cancer, there's not a single kind of ovarian cancer. There are multiple histological subtypes. Very familiar with this. Serous, endometrioid, mucinous, and clear cell are the four major histological subtypes that until about 10 years or so ago were all thought to arise from this single layer of cells that envelop the ovary called the ovarian surface epithelium. Right? Now, the most common of all these is the high-grade serous cancer. Unfortunately, this accounts for about 70% of all cases. This is the one we hear about when we hear about the silent killer and all the dire clinical associations with this disease. And so this is a slide that is essentially summarizing what Amy was talking about with regards to this new paradigm of where ovarian cancer starts. So it's been just about 10 years now since we published the first set of papers with Chris Crum at the Brigham, which was not the first time people had seen early tubal lesions in the fallopian tube. In fact, the Brca genes were cloned in the mid-90s, so 94, 95. By the end of that decade, we were offering women prophylactic surgery, and by early 2000, there were anecdotal reports of early dysplastic epithelium detectable in the fallopian tube of women who had a BRCA mutation in the setting of looking for an occult ovarian cancer. Okay, but it was sort of paradoxical. We're looking for ovarian cancer, and yet we're finding weird things in the fallopian tube. What's going on? And that's when Chris decided to systematically examine the fallopian tube. Because if you imagine my arm and my hand as the fallopian tube and the fimbria, what we used to typically do is take a section through the fallopian tube, show there was nothing there, and really we didn't look at the rest of it. We were focused on the ovary. I mean, it was almost incredible that we would kind of essentially throw it all out. And what we did with the SEE-FIM protocol was essentially say, we're going to look at the entire thing, we're going to focus on the fimbriated end, and we're going to see what we find. And this sort of summarizes a lot of those studies from about 10 years ago, where you have normal fallopian tube epithelium, you have ciliated and secretory cells. Those are the most common subtypes, most common cell types. And then you find this entity we call the P53 signature, which are stretches of secretory, benign-appearing secretory cells that stain very strongly for P53. And in small number of cases, especially in the BRCA population, about 5% of the time, you'll find what we call an intraepithelial carcinoma or a serous tubal intraepithelial carcinoma or STIC lesion. And then that can often be associated with the clinically manifest disease, which is the invasive high-grade serous, and they share a lot of these properties. There's detection of early DNA damage as measured by H2AX in these PP3 signatures. They actually do have mutations based on laser capture, microdissection, and sequencing of these entities to show that, in fact, the stabilization of the protein is due to mutation, and that same mutation is carried forward in any one given patient where you find these various entities. We've, not our group, but others, especially at Hopkins, have shown that you can demonstrate telomere shortening in these in situ cancers, which is a hallmark of all precursors. And copy number alterations have already been demonstrated to occur as early as the STIC lesion. So this is sort of what we believe represents the site of origin for majority, though not all, high-grade serous ovarian cancers, right? So I think more importantly is what do we do with that information now, right? I think most people who've looked have seen and have found these entities, right? So what are the consequences of figuring out that the fallopian tube is probably the culprit for a majority of these high-grade serous cancers that we call primary peritoneal carcinoma or tubal primaries or ovarian primaries? And I think they fall into two categories. One is prevention, and the other one is early detection. So for those who are familiar with this area, you've probably read a number of articles over the last handful of years trying to figure out what can we do about the fact that the tube is the site of origin. You'll recall that women who have a BRCA mutation get offered prophylactic surgery, tube and ovary removed, but that's associated with what we call a surgical menopause, right? All the complications of menopause are now sort of enacted instantly as opposed to traversing a number of years and gradually going through that process. And that includes cardiovascular and bone disease and a number of other. And so this is relevant because about 30 to almost 40% of women who have BRCA mutations refuse to undergo the prophylactic surgery because of that risk and that complication. They don't want to do it. And so the recommendations by the SGO are that women at risk, especially those that don't want to undergo the risk-reducing bilateral salpingo-oophorectomy, be offered a two-step reduction where you have interval salpingectomy with delayed oophorectomy, right? Take out the tubes first and then go back and take out the ovaries after they've gone through their normal menopause. There are two studies that are actually looking at this. The WISP study, Women Choosing Surgical Prevention trial, which is ongoing now, comparing the two, and the TUBA trial, both prospective studies that are actually looking at high-risk populations. The WISP study is being done at 6 centers across the country. I think the goal is to enroll about 300 BRCA mutation carriers and offer them this, and then obviously prospectively follow them and see whether over time their risk or incidence of cancer is similar to the bilateral salpingo-oophorectomy or not. I think we have to be cautious with this because for those who are involved in looking at pathology specimens on the GYN side, we're familiar with cortical inclusion cysts, which now people believe are probably coming from adhesions from the tube. Some surgeons who I've talked to say that oftentimes it's not so easy to separate the fallopian tube from the ovary, and that there can be really significant adhesions that leave behind tubal epithelium. All those things are going to really come into play in these studies to really determine whether, in fact, it's sufficient to take out the tubes, and under what circumstances and under what surgical approach is the best to be able to not only completely remove it, but be able to interpret that result as risk reduction or not. And for women who are at average risk, this term of opportunistic salpingectomy is really rising in its utility. So it used to be that removing the tubes at the time of hysterectomy was about a 5% incidence, and now in certain practices it's up to about 75%. And so we're basically saying take out the tubes if you're in there for fibroids, prolapse, abnormal bleeding, any reason where you're going to take out the uterus, take out the tubes. In fact, places like Vancouver and British Columbia have mandated this, right? So this is one of the things that you can do in Canada, and that whole province is actually mandated to actually take out the tubes when you go in there to do a hysterectomy for any reason. So those are the advances in terms of prevention that are ongoing, and I think it's going to be very interesting to see how this plays out in the next 5 to 10 years. In terms of early detection, Amy mentioned CA125. I mean, CA125 was discovered in 1983, so it's been a long time. The HE4 that you mentioned was approved 25 years later. So now we have 2 biomarkers that are not useful for early detection. So it's really been a struggle to really figure out, how to kind of look at this disease, because it's still the holy grail, right? I showed you those stats. You find them serendipitously at early stage, and you can make a huge difference versus the typical late stage presentation. We know that transvaginal ultrasounds are not in and of themselves adequate. The UK CTOX study that was published at the end of 2015, this was a huge study in the UK, 202,000 women that were stratified to look at observation or CA125 or CA125 plus transvaginal ultrasound at regular intervals. And what they concluded there is that they can detect early volume disease or recurrence, but it didn't make a significant impact on mortality, which was the primary endpoint. Though towards the end of the trial, this was a 14-year study, so they were actually monitoring women longitudinally for 14 years, they started to see some evidence that perhaps there may be some improvement in overall mortality. The study doesn't end until the end of next year, so we'll see what that analysis looks like in terms of that. But that's, again, looking at longitudinal. We're talking 15 years of screening, right? So we don't have a test that today you can just say, I think you have something. That's where there's a lot of excitement about the liquid biopsies. Most of you have heard about the excitement of looking at in the blood for evidence of early disease, and that can be not only looking at DNA, but exosomes, microRNAs, a whole variety of different entities that might be useful in that regard. And then a work I'm not going to get to talk about, but we wanted to look at this concept that the fallopian tube is the site of origin. So we did a study some years ago when I was still up in Boston, where we said, okay, can we find cases where a woman has a high-grade serous cancer, but it's limited to one side, which for any surgeon in the room, you'll know that doesn't happen often. But when it does, can you capture that other tube, and hopefully it's normal, and then compare what does normal fallopian tube from the same patient secrete versus the cancer. Look for things that are either difference in abundance or novel in the cancer that's not in the tube. And one of the things that we're working on now are what I call these binary markers, which are, we saw HE4 and we saw C125, so we knew we were kind of climbing up the right tree, but we also identified a number of proteins that are normally methylated and not expressed in normal fallopian tube. And in the cancer state, they lose that methylation and now they get re-expressed. And so that's exciting because the background level ought to be really low, right, in most women, and the question is it useful, and they're secreted. So we're pursuing those studies with the angle coming from the fallopian tube as the place to look. Okay, so fallopian tube, site of origin. Most of what we know genomically from this cancer comes from the studies of advanced disease in the Cancer Genome Atlas. This is mostly review. And what the TCGA showed us, so now this is looking at a number of different tumor types that were characterized by the Cancer Genome Atlas, right? So bladder, breast, colorectal, GBM, head and neck, etc., kidney, and here's ovary. You're looking at chromosome 1 down to 22. Red represents amplifications, blue represents deletion, and what you can make out is that ovary has the most complicated genomic terrain of any cancer that's been studied to date. There are more amplifications and deletions that affect every chromosome in the genome of ovarian cancer than any other cancer type. And at the bottom, you're looking at somatic mutation rate, and what you find is that ovarian cancer has really a scarcity of recurrent somatic alterations, right? The one mutation that is almost obligatory is mutations in P53, right? So mutations in P53 occur in 100% of these cases. In fact, the Cancer Genome Atlas reported 96%. But then Yimeng Shi and Bob Kerman at Hopkins went back and reviewed those 4%. Turns out they were low-grade serous cancers, so if you take those out, it's 100% of high-grade serous cancers have a mutation of P53. In the absence of that, you almost have to question whether you have the diagnosis right. So we were interested in trying to figure out how do you go from something like this, right? How does this happen? So we said, okay, can we move backwards in time and try to understand the evolution of this cancer, starting from the very early lesions in the fallopian tube that we identify, and move forward, trying to understand at what point do we see this sort of genetic chaos that occurs in these cancers. And so we did this in collaboration with Victor Velkulescu at Hopkins, and essentially we decided to collect a number of cases from women who had stage 3 ovarian cancer where we could identify all those lesions, right? So we had normal fallopian tube, the P53 signature, right? Benign appearing cells, the STIC lesion, the invasive fallopian tube cancer, the ovarian cancer, metastasis, however you want to look at it, and then various metastatic lesions in the peritoneal cavity, right? So we wanted to have cases where we had all that. We wanted to also get risk-reduced salpingo-oophorectomies from BRCA mutation carriers where the only thing they found was STIC, so in situ cancer without invasion. And the goal was to identify these cases that were relatively recently done in terms of how long they'd been in FFPE. And we had to figure out ways to do P53 immunohistochemistry on LCM slides. So if anyone's done LCM, you know these are metal slides. It's not typical to be able to do IHC on that and then do LCM. So we had to figure out how to do that. And then working with Victor to do whole exome and copy number analysis from minute fragments of tissue from which we extracted the DNA. And this just sort of demonstrates some of the LCM approaches and the types of lesions that we isolated from these patients. So I'm going to give you an example of some of the things that we found. So in aggregate, we have about 700 gigabytes of data that was generated from this with 115-fold per base coverage, which gave us about 45 alterations per sample, meaning every one of the tumors that we looked at had about 45 alterations in them. At the bottom, you're looking at P53 signature, the STIC lesion, the left tube. This was done before or after it was embedded in formalin to make sure that we weren't seeing differences. The right ovarian tumor, the rectal metastasis, the appendiceal metastasis, and the omental metastasis, right? And so what we find is that the earliest alteration is P53. Not surprising. It occurs in the P53 signature, and the same alterations preserved throughout. So we've known that for some time based on immunohistochemical studies, right? What this showed us was that, in fact, it's not the only alteration that occurs in these P53 signatures. There are other genes that are affected and mutated. None of them are recurrent, meaning we don't see them necessarily in other examples of this. They're private alterations, right? P53 being the only recurrent. So there's about 10 alterations that we can identify in the signature. When you look at the STIC lesion, you acquire another 10 to 15 mutations. And as you move away from the fallopian tube towards the metastatic invasive lesions, you acquire additional alterations. Right? Here's the omental met that has acquired a few more. But really, the STIC lesion actually has the bulk of the alterations that you see in the advanced disease. And this is not very different than the studies that were done in DCIS by Bob Weinberg years ago, showing that DCIS almost looks almost identical to the invasive lesion. This is at the time when they were trying to figure out what are the genes or alterations that actually provide that invasive potential. They really weren't able to really find it doing it that way because the STIC lesion or the DCIS was as complicated as the invasive cancer. Here's another example. Now I've sort of twisted this on the side. So here's the signature. There's 3 different STIC lesions we identified. The right fallopian tube tumor and the right ovarian tumor. And again, P53 is early and it's present throughout. These signatures have about 10 alterations. You acquire many more in the STIC lesions and then even more in the right ovarian tumor. So when you look at now not only at mutations but also LOH events, so copy number, allelic fraction changes, and now you look at 5 cases. So this gets a little busy, but here's the legend. Simply all the colors are showing you is not only the mutations but the LOH events that we see in every one of these entities. And again, even by looking at the LOH events, you see that the p53 signature here has the fewest. All the ones that are detected here are preserved as you move away from the fallopian tube, and you acquire additional mutations and copy number changes as you progress from non-invasive to invasive to metastatic sites, right? And that we see in all these examples. And these are just all arranged from signature to invasive disease from left to right. So if you now, and this is sort of the fancy bioinformatics stuff that I could not do it justice, but Victor uses this schism algorithm to look at the phylogenetic pedigree of these tumors. So you can actually start looking at the evolution of these cancers, from which lesion did each of these subsequent lesions emerge. And when we do that, we find that it's pretty linear in our study, right? So you start with the fallopian tube and the signatures. What we see is you acquire the mutation of p53 and a few others. You progress to the SIG and the STIC and the invasive cancer, and it really kind of goes from tube out, right? But I'm going to point out 3 things. One is that in all cases we see a mutation in p53, as I mentioned. So this is the truncal primary event that's obligatory in this cancer subtype. We find that most of them progress in this linear fashion, away from the fallopian tube and out towards the peritoneal cavity. But there's one example which I think is worth briefly mentioning here, which is that in this case, we had normal fallopian tube, we had the right STIC, we had the right ovarian tumor, and then there was a STIC on the other side. So histologically, right, the pathologist, us, looked at the other side, the contralateral side, and we saw what looked like a STIC, right? And so, there was bilateral STIC. But when you actually did the genomics, it turns out that this was a metastasis from the other side. Right, so it actually the STICs can masquerade as metastatic sites, and this is actually very consistent with a report that Ernst Langel just had at the end of 2016, where he saw the same exact thing. That in the setting of advanced disease, so when you actually have invasive ovarian cancer, there's involvement in the peritoneal cavity, a STIC could be masquerading as a metastatic lesion could be masquerading as a STIC because you can get involvement of the fimbriated end. They'll implant there, they'll grow there. And by our eye, we'll call this a STIC lesion. Yeah?
Did you guys, in that particular case, examine the endometrium at all?
No, we didn't. In our study, we didn't look at the endometrium. But it's...
I mean, in some ways, thinking about it, going back to breast, it's almost the same idea of those cells, whether you call it intraepithelial metastasis or simply transition throughout the entire epithelium, that they're actually migratory without actually breaking the basement membrane.
Yeah. Well, there's a lot of evidence that these STICs can start to slough off before they ever invade. And the question is, how did that happen? Right? Did it go sort of through the uterus, or did it come around? Right? It's a good point. And we hadn't looked at the uterus, but it's a cautionary note because I think in advanced disease, it's almost anything goes, right? And you have to be careful with that. So then it comes to the question of, are all STICs created equally? And the reason we asked this question was because of this concern about overdiagnosis and calling too many things carcinoma, and should we actually call these tubal intraepithelial neoplasms until we know about their biology, right? Is this a scenario, for instance, like CIN in cervix, where we have low grade and high grade, and then the low grades will oftentimes resolve and you just follow them, versus high grade, which we know the story behind that? And do we know enough about STIC lesions to know that they're all the same and they're all capable of invading and killing the patient, or perhaps there's some that are not? And so the way we wanted to go about this, and this is still pretty early, but we decided to compare the number of somatic alterations in STIC lesions that were identified in BRCA mutation carriers, that the only thing we found in them was an in situ cancer. So there was no invasive disease, and we compared it to STIC lesions that were present and studied in the context of advanced disease, right? So how many alterations do you see in STICs with associated invasive cancer versus STICs in isolation? And basically we found, it all falls within the same ballpark. There's about, this paper should be hopefully coming out, I say hopefully, in Nature Communications in the next few months. So we added about 3 more of these STICs alone, and they all fall within 30 to 70 or 80 alterations, which is kind of what we see with STICs that are associated with invasive disease. So at least at this one omic level, they're similar. Right? We haven't seen evidence that we can find STICs that are drastically different than those associated with invasive disease. So what are the take-home messages? I think the SEE-FIM protocol, which I didn't show you, but I think many of you are familiar with it, stands for sectioning and extensively examining the fimbriated end of the fallopian tube, is an important clinical tool for assessing risk reduction salpingo-oophorectomy specimens. I think precursors, high-grade serous cancers exist in the distal fallopian tube. The majority of them are associated with these STIC lesions, these in situ carcinomas, but as I mentioned, I think you have to be careful in the setting of advanced disease that a STIC lesion may actually be a metastasis, not necessarily the precursor. High-grade serous carcinomas are characterized by these early puberty mutations and then really widespread copy number alterations. And I think that's because you're losing the puberty checkpoint, and in many cases you're losing BRCA1, which is another DNA repair. And so I think when you lose those early, as opposed to later, because we know, for instance, in colon cancer, there's mutations in P53, but that tends to occur much later in the Vogelgram sequence of the evolution of this cancer, as opposed to being the essentially early lesion in ovarian cancer. So the genomic alterations here suggest that the precursors do exist in the tube and give rise to these metastases. And again, this sort of cautionary note about STICs in the presence of metastatic disease. And as most studies show, that the more we know, the more we still need to figure out and find out. And so what are the implications for early detection? I mentioned a little bit about that. What about prevention or therapies? And I think there's a lot of evolving studies going on and research happening right now to try to address those. I'm not going to stand here and tell you I have the answer to all that, but I will tell you about what we've been trying to do over the last few years. And as Amy mentioned, at the time that we sort of kind of started stumbling upon the fact that the tube was the site of origin, there were really no model systems to look and study the fallopian tube epithelium. We did a lot of searches. Most of what we found was in the reproductive biology realm, where you're trying to understand how is the fallopian tube acting as a conduit for sperm-egg interactions, fertilization, transport to the uterus. But there weren't model systems to look at the cells themselves as potential sites of cancer. So we had to sort of develop a lot of these model systems in the lab. And I'll talk a little bit about the mouse models that we've been working with, and then give you examples of what's happened in terms of model development and as tools, and also show some of the things that I think we've done that are great for collaborations if people are interested, and certainly for drug development. So mouse models come in 2 flavors. One is basically you take a tumor, you implant it in an immunocompromised animal, PDX models, right? And in a number of studies, including our own, people have shown that if you serially passage these tumors from mouse to mouse over many generations, remarkably they actually remain pretty faithful to the original patient tumor at the genomic level. So both by mutation and copy number alterations, we don't see drift or enrichment for certain subpopulations, which is a good thing. And then the genetically engineered mouse models are obviously in an immune intact mouse where we genetically knock out genes that we believe to be important in ovarian cancer, and that's informed by the Cancer Genome Atlas. So all the alterations that were described in the Cancer Genome Atlas, you look back and you say, okay, what goes together, what doesn't go together? Can we recapitulate some of this in an animal model? So here's an example of our PDX platform at Penn. We have 2 types of PDXs, what I call the primary ovarian PDX, or POP, and the metastatic ovarian PDX, or MOP. Fiona Simpkins, who's one of the junior faculty GYN oncologists in our center, developed the primary PDX model where she literally takes patient tumor and sutures it to the ovarian bursa in the mouse, and then closes the mouse and follows tumor development with ultrasound, and then serially passages these tumors from generation to generation. And our model was developed looking at the metastatic setting, where we isolate ascites fluid from patients who have advanced disease, isolate the tumor cells, and then put them back into the peritoneal cavity of a mouse, because that's the orthotopic site for advanced disease. And in many of these cases, we were able to luciferize these animals so you can actually monitor tumor progression after treatment with either standard therapy like carbo, Taxol, or the combination. And with bioluminescent imaging, you can follow the tumor response over time, and then the recurrence, and study resistance and various aspects. And we've genomically characterized these so we know which models are deficient in homologous recombination or DNA repair, basically the BRCA mutant PDXs, which are resistant to PARP, which is the exciting new drug, 3 of them now approved for ovarian cancer, and unfortunately many of them already are showing resistance in the clinic. And so what are the resistance mechanisms? We've developed PDXs to look at that. Cyclin E elevated and Cyclin E low, and I'll talk a little bit more about Cyclin E later on. So those are the PDX models. How have we moved on since I've been at Penn to implement them? And this is what I call our spectrum program. And the goal, the question we want to ask is, how do you go from a cell in the fallopian tube that is relatively well differentiated, Mullerian epithelial cell, to a cancer that kills you, right? From a tumor that initially is very sensitive to chemotherapy to one that is ultimately refractory to almost everything you try, right? And how does that natural history evolve? And I don't know of too many studies that have looked at this. David Botel published a really nice paper in Nature in 2015 where he started to look at these mechanisms of resistance in patients using a rapid autopsy program. And so what we've developed at Penn using a platform that is relatively new, based really on having our own tumor bank and our own PDX platform, is to harvest tissue at the time of diagnosis, follow these patients through their treatment, and then harvest, whenever possible, tissue at the time of recurrence, and then at time of death through a rapid autopsy program. And really try to understand at the genomic level, at every omic level you can think of, right? How do you go from something that's initially very sensitive to something that's initially going to kill you, looking at this over time? And so this is how we're trying to implement it. And in the process of collecting these specimens, we're not only characterizing them, but we're developing PDX models. So we actually have a animal surrogate or avatar, if you will, of the sensitive, resistant, and lethal cancer. So if anyone's interested in stuff like this, I'd be glad to talk more about it. And then the genetically engineered mouse models, I'll review what we know about that. And the first one actually was surprised everyone, because when we actually went back into the literature, we realized like in 2002 someone had actually hit upon this without knowing it. And so this model, it was out of a group in Japan where they were interested in understanding hormone-responsive cancers. And so what they did was they took the oviduct-specific glycoprotein, OVGP1 gene, and took the 2.2 kb of its regulatory elements in the promoter and then hooked it up to T antigen. So S340 virus T antigen is a transforming oncoprotein. It will inactivate P53 in an RB, and in many mouse models it'll transform these cells. And so now they had T antigen under the control of this OVGP1 gene. And when they induced T antigen expression, they got cancers all up and down the Mullerian system: fallopian tubes, oviduct, endometrium, upper endocervix, but not the ovary per se. And they did some studies looking at the hormone component, right? They did taking out the ovary, they blocked tumor development, suggesting there was hormone responsive, and they identified the elements in the OVGP1 promoter that were responsible for this. And that's all fine and good, but the reason that I mention this is because they not only did they get tubal cancers, but about 12 years later, Pat Morin stumbled upon this paper and said, hey, now in the context of what we know about the fallopian tube, why don't we go back and carefully examine the fallopian tubes of these animals? And what he found was that he could identify P53 signatures and STIC lesions in the fallopian tube that essentially mimic what we had seen in the human setting. So exactly same model system, but now demonstrating that, in fact, transformation of the fallopian tube was undergoing a very similar process. Now, Kathy Cho at University of Michigan has taken the same model now one more step, right? So now what they find is that if you look at just those 2.2 kilobases of the promoter, I told you get cancer all up and down the Mullerian system. What she then said is perhaps we're missing some of the regulatory elements because this particular gene is very restricted to expression in the oviduct. They don't see expression in the endometrium. They don't see expression in the ovary. So why are we getting cancers everywhere? Maybe we're missing some of the regulatory elements. So she actually cloned all the regulatory elements on a bacterial artificial chromosome, a BAC, and has a BAC driving a tamoxifen-inducible CRE. And so now you can actually knock out whatever genes you want under very exquisite regulation in the oviduct, right? And this is a paper. She had one of the stories recently came out in journal Pathology. The other story should probably be coming out there as well within the year, looking at the various genes that contribute to the development of these cancers. Another model was reported by Marty Matzuk, where they knocked out P10 and dicer using the anti-Mullerian hormone receptor type 2 in a mouse model. I think that they were probably trying to study aspects of uterine biology, but what I think they stumbled on was a tubal phenotype. So they got tubal cancers. When they removed the fallopian tubes in the mouse, they blocked tumor formation. But this model does not take into account that PP3 is an obligatory event in this disease, and it's an early event. Nonetheless, the histology looked pretty decent, and they got a tubal phenotype. And then the model we reported a few years ago was based on a transcription factor that I think is fascinating called PAX8. So PAX8 is a member of the PAX family of transcription factors that's involved in development. Different PAX family members control development of different organs in the body. And if you knock out PAX8 in a mouse, you block formation of the female reproductive tract. So you get atresia of the uterus and the tubes. Occasionally it gets hydrosalpinx of the tubes, but you don't actually form a female reproductive tract. The ovaries are perfectly fine. So it's essential for the development of the Mullerian system. It's retained in the adult secretory cell, which is what you're looking at here, ciliate cell, secretory cell. Nearly 100% of high-grade serous cancers express PAX8 to some degree. So you need it during development. It's retained in the adult state, and it's important in cancer because if we knock it down in ovarian cancers, they die. We trigger apoptosis. So it seems to be one of these transcriptional master regulators, lineage-specific transcription factors. And so we thought, not only is this interesting, we've published studies looking at where in the genome does PAX8 bind to start to understand how does it do something that dramatic, right? What are the genes that it controls, and how does that systrome change between normal and malignant cells, right? So we've been interested in PAX8 for a while, but we also took advantage of this to generate a mouse model where we actually had the PAX8 promoter driving the Cre recombinase, right? So now you only had deletion of whatever gene you're interested in PAX8 positive cells, which in the mouse are really highly enriched in the fallopian tube. And so this is just a summary slide where we saw by knocking out PP3, BRCA1, plus or minus P10, we could recapitulate the emergence of STIC lesions that looked a lot like the STIC lesions we had described in humans in invasive disease. But the question that we were most interested in, and the caveat that a lot of people raise with mouse models, is how well does your model recapitulate the genomic complexity of the human disease, right? And I told you ovarian cancer is characterized by widespread, disaster essentially throughout the entire genome. So how well did our model recapitulate this? So we looked at a number of these cancers by array CGH, comparative genomic hybridization. And so here what you're looking at is chromosome 1 all the way to the end here in 3 different tumors. And blue in this case is amplification, red is deletion. But what you can see is that we saw amplifications and deletions throughout the entire genome of these animals, even though we only altered 2 or 3 genes. Right? So we saw amplifications of MYC, which is the most commonly amplified gene in high-grade serous ovarian cancer. We saw amplifications of KRAS, which is actually not mutated in high-grade serous cancer, but is amplified in human disease. We saw loss of INPP4B, which is a tumor suppressor that Luke Cantley has been studying in the context of PI3 kinase pathway regulation, and that's lost in these tumor types. And so it was actually quite nice when we actually compared the top 20 or 30 genomic alterations from this model to their homologs in human. It actually matched up pretty amazingly with the frequency of alterations that we saw in the cancer TCGA. So it looked like we were able to recreate the genomic complexity of this disease by targeting the right cell with the right combination of genetic alterations. And so we in all these studies, including ours, we can target the fallopian tube secretory cell in vivo. When we do it, we can recapitulate the pre-invasive and invasive lesions that characterize a human disease. The STIC lesion in this case is in fact a precursor. If we do oophorectomy in these mouse models, you don't block development of the cancer. You still get transformation of the fallopian tube. But if you take out the tube, you block tumor development. The interesting thing with that, and we found it in our study, was that even though the ovary is not the site of origin, if you take out the ovary, again, you still get transformation of the fallopian tube, but now the tumors don't disseminate to the peritoneal cavity as readily or as widely as when the ovary is there, suggesting that perhaps the ovary is an important sort of first stop, seed and soil perhaps, that enables the tumor to grow and then progress from there. And that's an area that a lot of people are trying to understand further now. These mouse tumors recapitulate the genomic complexity of this disease, and again, as I just mentioned, oophorectomy does not block STIC formation, but it has an impact on dissemination of disease. Okay, so how do we utilize all this to really attack these tumors more effectively? And this is what I'll try to talk to you a little bit about for the rest of the talk. So we went about this trying to do what not everyone else is trying to do, because there's way smarter people doing that stuff. So we decided, let's see if we can attack an aspect of disease that is not necessarily, in the main light right now. And so if you look at the Cancer Genome Atlas results, right, half of these tumors are what we call HR, homologous recombination deficient. They have mutations in the germline or in the somatic genome for BRCA1 or 2. You can even have methylation of the BRCA1 promoter that essentially wipes out HR. You have deletion or amplification or loss of other HR genes in the pathway, including the Fanconi anemia pathway. And if you add that all up, about half of all high-grade serous cancers have a defect in HR. The prediction from that is that these are the tumors that are going to respond to DNA-damaging agents like cisplatin and PARP inhibitors, right? The other half of the pie is actually HR proficient. These tend to be chemorefractory tumors, and so far are characterized by cyclin E amplification, RB, or NF1 loss. And this is from recent study from David Botel's group. We were particularly interested in cyclin E amplifications because it's one of the most common amplicons in high-grade serous cancer. It occurs in about 20% of these tumors. If you look at the genome, again, of ovarian cancer, again, chromosome 1 all the way to 22, amplifications in red, deletions in blue, and again, you see that there's a lot of genomic disarray in these tumors. Here's cyclin E on chromosome 19q12. Okay? So it's the third most common amplicon in this particular disease. What does cyclin E do? The normal function of cyclin E is to partner with its kinase partner CDK2, cyclin-dependent kinase 2, to govern that transition from the G1S phase of the cell cycle into the S phase of the cell cycle. When you have too much cyclin E, you get what's called unscheduled S phase entry. You're pushing the cell into S phase before it's ready. Okay? That creates a lot of replication stress, DNA replication stress, and oftentimes you can see centrosome amplification as well. So Alison Karst, who is a really talented postdoc in the lab, decided to ask the question, well, if cyclin E is important, how early is this occurring in our now new understanding of high-grade serous ovarian cancer development? And to begin to look at that, she used a tissue microarray that we had developed at the Brigham, which was a collection of about 150 high-grade serous cancers for which we had the primary tumor, the metastasis, and some lymph nodes, and used FISH to look at copy number for cyclin E. So she used a probe that was specific for cyclin E and a control probe to look for the ratios to define gain and amplification, and this was done independently by cytogeneticists, so we were not involved in that. And you can find that about 25% of these cases have a bona fide amplification in cyclin E. If you then do immunohistochemistry on the same TMA, you find that about the same fraction has high levels of cyclin E expression. So the more cyclin E copies you have, the more protein you have. So then she said, okay, now let's go back to our paradigm. Normal epithelium progressing through these precursors to highly invasive ovarian cancer. At what point do we see dysregulation of cyclin E? So we looked at p53 signatures. Again, you can't really make it out of this power, but here is a stretch of benign-appearing secretory cells that stain very strongly for p53 but do not proliferate as measured by Ki67. And yet in these cells we see expression of cyclin E, and this was in about a third of all cases. If we then look at STICs, the in situ cancer, this is high-grade serous in situ disease that's positive for p53. It's proliferative now, and it still expresses cyclin E. And in fact, with Yimeng Shi at Hopkins, we recently did FISH on these sections and can show that you can see by copy number and FISH amplification of cyclin E in the STIC lesions, right? So again, it's happening early on. So that's when Alison said, okay, can we go back now to our sort of battery of model systems and ask, can we sort of model this in the lab and figure out what cyclin E is doing? And so this is a schematic of the models that we've been developing. So I told you a little bit about our PDX platform. I described some of the genetically engineered mouse models that have evolved. We've also developed a battery of fallopian tube secretory cells. So this is what we think are the cells of origin that we've immortalized in the lab that enable us to ask what's the contribution of any genetic alteration on the ability to transform these cells. And we also have a platform to look at what I call the ex vivo fallopian tube platform, which is essentially primary cells isolated from the fallopian tube, grown in these transwall filters where they retain their polarity, and you can ask various questions regarding what they secrete, how they respond to genotoxic stress. So Alison went to our cell line model and asked if we isolate cells from the fallopian tube, immortalize them in culture with telomerase and a p53 mutation, which is obligatory since all these tumors have a p53 mutation, overexpress cyclin E or vector, what happens? So panel A, basically you see cyclin E is overexpressed by Western blot. It activates its CDK2 partner as measured by phosphorylation of this protein. Essentially everything else stays the same, CDK2, PAX8, CDK7, but the cells become more proliferative. That's not too surprising. They also become clonogenic, and they lose contact inhibition. Most epithelial cells, once they grow and start touching each other, they stop growing. These cells start to pile up on top of each other. But they don't really form colonies under anchorage-independent conditions very well. And anchorage-independent growth is sort of a marker of transformation in culture, right? So we didn't really see strong evidence for that. And I'll come back to that. But what you did find was that when you overexpress cyclin E, you get more DNA damage. And this is sort of looking at H2AX, that histone that gets phosphorylated at sites of double-strand breaks when you have a lot of damage. And you can also see this by COMET assays, where you actually look at the degree of fragmentation of the genome by running these cells under electrophoresis conditions. So too much cyclin E causes replication stress, causes DNA damage. And when you profiled these cells, this is now a volcano plot, where you're looking in the top right here at the genes that are highly overexpressed when you have too much cyclin E. Right? The genes that are highly overexpressed are genes all involved in DNA replication for protection in the BRCA1 Fanconi anemia pathway, interstrand cross-link repair. And we could verify this by Western blot, where you can see that FANCD2 and XRC2 are overexpressed at the protein level in the cells that have cyclin E. So too much cyclin E causes DNA damage. The cells upregulate the DNA repair pathway. Okay? Why is that interesting, or actually fascinating? It's because of, well, so here's validation by RNA-seq. So we just confirmed this by RNA-seq. High correlation with our nanostring assay. And if you look at the TCGA, in genes that have cyclin amplification, they correlate most highly with Fanconi anemia and HR pathway. Okay, so there's something to this upregulation of these DNA repair pathways. And the reason it's really exciting is because David Botel published a paper shortly around the same time where he did a synthetic lethal screen for cyclin amplified cells. So what does that mean? He said he took all the cell lines that had amplifications in cyclin E and did a genome-wide RNAi screen, asking what genes, when you knock them down, kill cells that are cyclin amplified. Right? And the genes that came up are the exact same genes that we showed were upregulated when you overexpress cyclin E. CDK2, its kinase partner, FANCD2, XRC2, CDC25, and BRCA1. So BRCA1 is synthetic lethal with cyclin E. And that's why in the Cancer Genome Atlas study that was published in 2011, they made the observation without the explanation that you never see cyclin amplification with BRCA1 mutation. They're mutually exclusive events. That's because they're synthetic lethal. Right? So we were very excited by this observation, because now it turns out over too much cyclin E, you upregulate the repair pathways, and once you do that, you cannot take it away. The cell depends on it. If you take it away, it dies. There's genomic catastrophe, right? So that kind of still brings us back to how do you target these tumors, right? So as I mentioned, the partner for cyclin E is CDK2. CDK2, CDK inhibitors are actually in the limelight quite a bit now. And so we wanted to ask the question, well, how dependent are these cyclin amplified tumors on CDK2 and cyclin E? So here are 3 ovarian cancer cell lines that have amplification of cyclin E. Here's one line that has a gain in the locus, and here's 3 lines that are not amplified at all. And when you knock down cyclin E or CDK2 in the amplified lines, most of them die. The other cell lines could care less. So there's a dependency on cyclin E and CDK2 in the amplified lines. So the question then becomes, if we now go to our library of CDK inhibitors, and many of you are familiar with the success of palbociclib in breast cancer, are there any CDK1 or 2 inhibitors that could mimic what we're seeing when we knock down CDK2 or cyclin E in high-grade serous cancers? And dinaciclib is one drug that we're interested in because it's under development by Merck. It's sort of been on and off development, but one of the better ones. And so we did a study looking at the amplified gain or unamplified lines, looking at their amplicon-dependent sensitivity. And unfortunately, we didn't see any. Right? So we didn't see any difference in the amplified lines, how they respond to these CDK inhibitors. And I think a lot of that is due to the fact that these CDK inhibitors are not specific. They hit a lot of other CDKs. You hit the transcriptional CDKs. I think that's part of the reason why they're associated with so much toxicity in the clinic now. So then we did 2 things, and we'll wrap up hopefully on time with that. we did a small molecule screen to identify compounds that could synergize with dinuciclib. So if we reduce the dose of dinuciclib, could we find a small molecule that will synergize with it? Okay. And at the same time, we did a gain-of-function screen to identify events that would cooperate with cyclin E. Remember, I just told you that cyclin E alone was not enough to fully transform these cells. It stresses the cells, it causes damage, but it doesn't fully transform them. The question was, can we identify genes that will cooperate with cyclin E and now fully transform them? So the screen that basically was run out of the Walter and Eliza Hall Institute in Melbourne, Australia with David Botel was a 4,000 compound screen, a very targeted screen that hit a lot of transcription, epigenetic, kinase factors. We titrated all the EC50s for all these, did this in an automated way, and then looked to see whether we could see synergy with dinuciclib in cell lines that were amplified for cyclin E versus cell lines that were not. And long story short, the compound that popped out was an AKT inhibitor, MK2206, another Merck compound. And we could see here that if you use Cho-Talele criteria for synergy, you find that the amplified lines showed strong synergy with dinuciclib and this AKT inhibitor, and the non-amplified lines did not. So there seems to be some connection there. In fact, if we use two different AKT inhibitors, the MK2206 or the GSK inhibitor, again, we see that the amplified lines show synergy and the unamplified lines do not. So then we took this into animals. So these are xenograft studies where we took OVCAR3 cells and planted them into animals, treated them with either single agent of each or the combination, and we find that the combination essentially causes tumor stasis or regression, whereas in the unamplified lines, they didn't really seem to care, suggesting that there's a sensitivity in amplified lines for this particular drug combination. We've recently gone back and looked at our PDX platform to identify PDXs that have the amplification for cyclin E. This is one example. Kai Doberstein in the lab has done this experiment where you now take these bioluminescent PDXs, you compare it to carbotaxol, which wipes everything out, and to the combination. And what we find is that you get a response to the combination. You stop treatment, the tumor will come back, but it stays sensitive if you retreat. So we're still working out those conditions. And in fact, we're in discussions now with Merck to see if we can convince them to do a study with dinuciclib and their MK2206 inhibitor. So going now back to the gain-of-function screen, and you'll see how these two come together. Alison looked at these cells, and instead of doing a genome-wide gain-of-function screen, she said, let's look at all the genes that are amplified in ovarian cancer. There are about 63 recurrent amplicons in ovarian cancer, which constitute about 1,000 to 1,100 genes. So she said, instead of doing a whole genome screen, let's just take those 1,100 genes and ask which of those will cooperate with cyclin E. And so the way she did this was, here's her open reading frame library of all those amplified genes. She puts them into her cell lines that had p53 and cyclin E and asks for in vitro ankyrogen-dependent growth. She did it in two ways. She looked at ultra-low attachment plates and soft agar, which is sort of the gold standard, mostly to see if there's overlap and what the differences are. And so these are the results. There is some overlap between the two, but there are some genes that pop out in one and not the other, and we're following up on a lot of these now. I'll point out the fact that AKT came out of the screen in the soft agar assay, AKT1 and AKT3. I'll tell you now that AKT2 is amplified, was not in this particular library. It was incomplete library. But all three AKTs came out of that screen. So Kai went back, took our fallopian tube cells, and overexpressed either AKT1, 2, or 3, and asked what is AKT1, 2, or 3 in the setting of cyclin E overexpression due to these cells, and was able to show under clonogenic, but mostly under anchorage-independent soft agar conditions, that AKT2 and 3 are very competent at transforming these cells in vitro, again, showing that they have a cooperativity, and he was essentially able to confirm this in the validation study. So why is that interesting? So David went back and said, okay, so what's the connection between cyclin E and members of the AKT pathway, right? PI3 kinase, AKT, all the members, mTOR, etc. So he looked at the cancer genome atlas. Here's the cyclin E amplified in 20-some-odd% of these high-grade serous cancers, AKT1, 2, and 3, and asked which of these are actually statistically connected to each other. And the only one that came out of that was AKT2, not the other two. The reason that it's AKT2, which I thought was kind of cool, was the fact that it actually is on the same chromosome as cyclin E. So when you amplify Cyclin E, you take AKT2 along for the ride. That helps you transform these cells, but you also create a dependency and a therapeutic opportunity. So with the small molecule screen, we picked up AKT inhibitors. With a gain-of-function screen, we picked up AKT as a cooperating event, again suggesting that there's cooperativity but also dependency once that cooperativity is established at the genomic level. And so we're, again, working with Merck trying to see if we can bring this to the clinic. So in summary, cyclin E amplifications are common in high-grade serous cancers. They cause replication stress and DNA damage at the replication fork. These genes that are upregulated to tolerate that stress then become synthetic lethal with cyclin E. And in genetic and chemical screens, we have identified AKT as a therapeutic target in these cells, and we're trying to work on clinical trials for that. But you'll remember, cyclin E amplification in ovarian cancer is only 20%, right? So why are we so excited about this? If you look at the Cancer Genome Atlas and ask across all the tumor types that have been studied to date by the TCGA, and that's a lot, right? What's the status of cyclin E amplifications? Amplifications here indicated by red. You'll see that uterine carcinosarcoma, very high. Almost 45% of them have amplifications of cyclin E. High-grade serous cancers right next to it. Bladder, stomach, etc., all the way down to triple-negative breast cancers have reasonably high levels of cyclin E amplification. So the idea is if we find something on our end that works, perhaps it could be applied to other tumor types. And just to give you an idea, the recent paper by Doug Levine's group looking at uterine carcinosarcomas, which have a high level of cyclin amplification, confirmed the high levels of cyclin amplification in these tumor types. And we've started to look at these tumors. Here's an example of a triple MT where you see strong cyclin E expression, and at the copy number level you can see amplification of cyclin E in this particular tumor. So we're hoping that what we learn by studying cyclin E in this particular setting of ovarian cancer might have applications to some of the other tumor types that exhibit this amplicon. And with that, I will, since we're in a pathology department, I will histologically acknowledge all my colleagues. These are former students that were with me at Dana-Farber, and my the new colleagues here, the old colleagues there, my new colleagues at Penn, and my colleagues at the Brigham and the Farber, and others I've had the pleasure of working with over the years. Thank you for your attention.
Thank you for the very educational presentation. You hinted to the role of the ovary in transitioning STIC to disseminated peritoneal disease.
Yeah.
It would be interesting to hear your experience and view on the theory that diagnosing STIC even in prophylactic PSO would put the patient at risk for developing primary peritoneal serous carcinoma, and whether these patients, in your experience, need follow-up.
Yeah, it's a good question. And I think it goes back to the issue of how much do we know about these STICs. Someone was asking earlier about their ability to disseminate before they ever invade. There are a number of studies that actually show sort of these sloughing off cells before there's any evidence of invasion. And so you can take out the tube. And that's why I think we have to have caution. I mean, it's very exciting to think about changing the paradigm of prevention without having to put women through surgical menopause, right? But I think we have to be careful with the idea that when you can, I mean, one thing is to take out the tube, find nothing. That's good. I think if you take out the tube and find something, you really have to reconsider that option and consider taking out the ovary as well, because it does seem to have a propensity to bind to and stick to the ovarian surface, much like normal fallopian tube fimbria can adhere to the surface of the ovary under normal conditions, right? So I think it's something we have to be careful with. I think if we find nothing, then that's an interesting study to follow up and see risk over time in BRCA mutation carriers. But in the setting of something, I mean, there's two studies I'm familiar with, one from Beth Carlin's group and one from Chris Crum, that looked at short, moderate follow-up in patients where all you had was a STIC lesion. And so they looked, I think, 5 or 7 years later. And if you only had a STIC lesion, I think it was 1 out of 15 recurred with primary peritoneal. If you had any evidence of invasion, and I'm talking like any evidence of invasion, it was 7 out of 15, right? So, what's that window of opportunity, right, between finding a STIC and an invasive disease? And is that really the precursor we want to target, or is the signature the thing we want to focus on? And I think we still don't know. And we're doing all these studies now. It's a good question. Yep.
So a number of labs have shown that PI3 kinase AKT activity actually supports BRCA, specifically BRCA gene expression. I'm wondering, have you considered trying, specifically in the cyclin E amplified tumors, actually just leaving CDK inhibitors out of it, use the AKT inhibitors, and go back to some of our standard platinum agents that actually those particular tumors are initially not responsive to, but in the context of if you actually use an AKT inhibitor where it's basically addicted to that, could you induce that synthetic lethality in the context of a DNA damaging agent?
So we've done it with, like I showed, with AKT and dinocyclib. With standard therapy, I have to go back and say, I don't think we've done that. So not in the PDXs. Right? I mean, ultimately, we try to go back to the PDXs on that, but I don't think we've done that yet. We've looked at PARP inhibitors and WEE1. ATR seems to be actually a really interesting target in that most of these tumors are also addicted to ATR. And so we're seeing really nice combinations with that. But that's a good point.
More questions?
Yep.
Is there any evidence that use as carcinomas of the ovary arise from fallopian tubes?
Good question. So I think Bob Kerman and Chris Scrum have small studies that have shown you could find mucinous sticks in the tube. But I think that's about the only evidence so far. I think our understanding of mucinous carcinoma in terms of how many are truly ovarian primary versus appendiceal, I think that's sort of evolved a lot, and whether there's a progression from these early to late stage ones, that's changed quite a bit. I think the frequency of truly primary ovarian mucinous tumors is lower than we thought. Someone else had their hand up. Yeah.
That was a great presentation. I was wondering about sort of if you sort of move forward with the clinical trial, because we've got you mentioned this initial trial with CDK4/6 inhibitor in recurrent ovarian cancer, and it's essentially flat out negative. We tried to stratify based on RB and P16 expression, and that didn't unfortunately pan out.
Cyclin E.
My question is, would you do an unselected trial, or you would enrich for Cyclin E?
So Dennis Slayman is involved in that study, and one of the resistance mechanisms of palbociclib is cyclin amplification. So we're actually working with Gottfried Konecki at UCLA and Dennis to look at that potential opportunity in that recurrent resistance setting, because it seems to uncharacteristically pop up CDK2 cyclin E as a resistance mechanism to CDK4/6 inhibition. It's a good question. Any others? Any donuts left?
Yeah.